Okay, for Unit 2 with bacteria pathogenicity, you guys selected several virulence factors, pathogenicity factors to focus on, and one of those was toxins. So in this audio lecture, I'm going to provide some of the background information, some additional detail about exotoxins and endotoxins, just in case you didn't get that section of the paper. And these are for the most part, these are lecture slides that are included into that Chapter 25 lecture slides. So I've just kind of pulled those out to give you some more information, what I would traditionally lecture on if we didn't actually cover that review paper. So when we look at toxins, we want to keep in mind the reason for having a toxin can be actually to subvert the host, and so to have an effect on the host, so it makes it more likely for the pathogen, the bacteria, to become a pathogen to cause disease. And so there are two main types of toxins. There's exotoxins and endotoxins. Exotoxins, exo, secreted out, toxin, lethal, can kill a cell. And so exotoxins are proteins which are produced by the bacteria. These tend to be small proteins, and they actually get secreted out. So we need protein synthesis to occur, and we also need secretion to occur. So for exotoxins, this is a very active process for the bacteria to produce the exotoxin and then again secrete it out. And then if the host cell has the correct receptors that can bind that protein, that exotoxin, the cell will actually be affected. Now, if you kill the host cell, those nutrients, the components inside the host cell, are now available for the bacteria to utilize itself. So again, exotoxins are small proteins, and they would have to be produced by living bacteria, because those living bacteria have to be present in order to undergo protein synthesis and secretion of these small proteins that act as exotoxins. For endotoxins, endotoxins are found within within toxin, toxic. So part of LPS, or lipid polysaccharide, which is found on gram-negative bacteria, can act as an endotoxin. So lipid polysaccharide is made up of three parts. There's a lipid portion known as lipid A. There's a saccharide portion, a sugar portion, and then there's a portion that's known as O antigen. So O antigen, immune system. O antigen can be seen by can give rise to an immune response, and this actually leads to the release of the lipid A. Now, lipid A can get incorporated into any cell that has lipids. So I'm going to give you a moment to think about what cell would have lipids. And so the answer would be any cell, right? So all cells have a phospholipid bilayer, and so this lipid A can actually be incorporated into any cell. And so with the exotoxins, we saw that I just said how it was very specific. You had to have the correct receptors for it to then get in. With endotoxin, this is a very non-specific toxin, so it can have a profound effect on any cell. Now, we're going to talk a little bit, compare and contrast exotoxins and endotoxins in just a couple slides, but just kind of keep that in the back of your head about the differences between exos and endotoxins. Okay. So exotoxins, again, they're proteins, and so proteins can have different effects on a cell. And so when we look at the mechanism of action, or how the exotoxin actually affects the host cell, it would be like our cell, there can be many different ways in which it can affect it. So as we can see from this list on this slide, we can have anything from plasma membrane disruption, and so very much like a detergent where it gets incorporated into the plasma membrane and then the cell can get lysed. We can also have effects to the cytoskeleton. So if we're talking about a eukaryotic cell, that cytoskeleton is very important for maintaining the shape and structure of the cell. So if we disrupt that, we're not going to have the cell being able to function as it normally would, because it's not going to be able to maintain its shape and structure. Protein synthesis, so obviously if we disrupt that with an exotoxin, the host cell, the eukaryotic cell, wouldn't be able to make proteins, and so they then wouldn't be able to function. So as you can see from the list, there's a number of other cellular processes that it would affect. And I just do want to point out that exotoxins don't just affect eukaryotic cells, that the bacteria are producing exotoxins to actually combat or fight with other microbes. And so sometimes the casualty would be humans, right, their host. So this figure from your textbook shows you several different classes or mechanisms in which exotoxins can be utilized. And as you can see in the middle panel, panel B, we have inhibition of protein synthesis. So the Shigella toxin would affect the ability for the ribosome and the messenger RNA to function and add amino acids on at the end of the peptide. As I mentioned with endotoxins, this is where we're looking at LPS. And so you may remember that gram-negative bacteria have an outer membrane. And that outer membrane is what prevents crystal violet from entering into the gram-negative bacteria and giving you a purple staining for a gram stain. And hence why a gram-negative bacteria ends up taking up the counter stain instead. So that outer membrane has a number of molecules in it, and one of those molecules is lipid polysaccharide, or LPS. As I mentioned, lipid polysaccharide is made up of 3 subunits. So there's going to be the O antigen, there's going to be our saccharide, and then we're going to have our lipid A. And so that lipid A can get released when the immune system attacks the bacteria and kills the bacteria. When the bacteria dies, just because of a normal, there's a normal amount of death that's going to occur in the bacterial culture or in an area where we have an infection. The patient could be taking an antibiotic, the correct one that kills the bacteria, and so in all these circumstances, lipid A would get released. And again, lipid A can get incorporated into cells that have lipids, which would be any cell, right? And so instead of having a specific cell that it's targeting, any cell could be affected. And so this is what leads to kind of a systemic effect with endotoxins. So endotoxins can cause fever, septic shock, and even death. And because it's a lipid, we really can't make antibodies to neutralize them. So with exotoxins, we could have antibodies that actually neutralize the exotoxin because it's protein. Well, with a lipid, we don't make antibodies against lipids very well. So the ability to neutralize it doesn't work well. And then again, because it's not affecting just one organ system but multiple organ systems, it becomes an issue. And so with gram-negative infections, previously it was about a 50-50 shot in whether if you had a clinical infection where you were actually going to a hospital because the infection was that severe, that you would actually survive. The statistics are actually becoming worse, so it's about 60% mortality rate with these severe gram-negative infections, again, where you are clinically showing symptoms. And so that also has to do with antibiotic resistance that we're seeing with the gram-negative bacteria. Okay. And again, the important piece to point out is that the bacteria is dying. So with endotoxin effect, you actually do need to have a high number of bacteria present to have the effect. With exotoxins, because it's protein, it's very specific, you actually don't need as much. So this is a table that I took from a different textbook because it really nicely summarizes up the differences between endotoxins and exotoxins, and a lot of which I've already talked about in the previous slides. Important pieces to keep in mind is the chemical composition, and so with exotoxins, again, these are small proteins. With endotoxin, this is where we're looking at that lipid A, the lipid polysaccharide that's found in the outer membrane. When we look at the amount that's needed, so toxicity, which is the first row, with exotoxins, we just need small amounts because it's very specific. It's targeting a specific type of cell. With exotoxins, or for endotoxins, we actually need larger amounts of it, but we can have a very systemic effect. So it's not isolated to a single cell type, but any cell. And the other thing to point out is heat. So the fourth row, heat denaturing, at 60 degrees Celsius. Because exotoxins are proteins, they're less stable, so they're very unstable. So if you have something like a fever, you actually destabilize the exotoxin. For endotoxin, because they're lipid-based, they're much more stable. So even with the fever, even though it stimulates a fever, you don't always end up getting the same relief that you would from the toxin. And lastly, typical sources, which is the last row. With exotoxins, these are gram-positive and gram-negative. With endotoxins, it would only be gram-negative. So that concludes the slides for our toxin audio lecture. So hopefully you have found this helpful, but I also highly recommend reading through the textbook and also that review article that we had the group activity on. So thank you.